Welcome back to Monkeys on Typewriters on YouTube. And if you've watched the previous episode on the evolution of cancer, which was part two, and where Arijit talked about the ways in which viruses can be involved in the origin and progression of cancer, you may have got the feeling that we were heading towards a certain discussion, a certain virus that may show signs of doing similar things. And in fact, that is what tonight's topic will be about. So we're going to have the next presentation will specifically focus on SARS-CoV-2 and whether it has properties that make it a plausible carcinogen, like the ones we've talked about in the previous episode.
Great. Thanks, Ryan, for the introduction. So today we're going to talk about this topic. And before we get into it, let's just say, right, there's lots of different things that are carcinogenic. Sunlight is carcinogenic. Red meat on the grill is carcinogenic. Beer is carcinogenic. Most of us don't think twice about throwing sausages on a grill and then washing them down with beer on a sunny afternoon. So it's not a big deal if something's carcinogenic, but if you know something's carcinogenic, then you can rationally optimize your risk and you can rationally optimize your approach towards it. So what we're trying to do today is not so much like spread, FUD as the minimizer community says. This is not about spreading FUD, fear, uncertainty, and doubt. This is more about asking the question, is this a plausible risk? And if so, if it is plausible, we'll go into in the next episode, we'll talk about if it was plausible, what kind of evidence would we need to buy into whether or not it was plausible. And we'll also talk a little bit as we go into this further into the series about what does that mean if it's a plausible risk? Sunlight is a carcinogen. As I said, no one's advocating that you go find your parents' basement if it still exists and live out the rest of your days there. We all face sunlight, but we use sunscreen. So if SARS-CoV-2 is a carcinogen and we know about it, we can deal with it differently than if SARS-CoV-2 is a carcinogen. And the first warning sign that we have is that it's a carcinogen is a lump on our, in our neck that we weren't expecting to have. So that's the distinction. It's a difference between knowing you're doing something that's risky and doing something that's risky without knowing it. So in that spirit, let's talk about this a little bit. And this has been a big rambling, 3-part, 4-part series. So it's been a while if you watched the first part. So we'll do a little bit of a recap of where we were and really talk about this, take it from the top. And so what we had talked about in the first part was that there are different paradigms that shape science, and these paradigms have a big impact on how we see the world. So science doesn't just proceed by looking at data and saying, aha, that's what it is. Instead, what science does is it operates on the basis of paradigms. So if you believe that the sun revolves around the earth versus the other way around, you're going to see things differently. And when you see things differently, every piece of data that you get, you'll be able to incorporate into your worldview. Similarly, with cancer, the dominant worldview is that cancer is a disease of oncogenes. Now, that dominant worldview of cancer being a disease of oncogenes is dated at this point. The evidence supporting that worldview is actually quite weak. And we talked about that at length in episode 1. If you want to go back and recap on that, go ahead. But a more clear picture is emerging of how cancer behaves, how cancer is initiated, and how cancer progresses. And the vast body of work that's been done in the last 20 years or so supports quite strongly the idea that cancer is actually an evolutionary disease. And by that, I mean that cancer is not many different diseases, it's one disease. So it's a process of somatic Darwinian evolution that involves a stochastic accumulation of mutations. And that stochastic accumulation of mutations happens simultaneously or in parallel in many different cells within the tumor or within the cancer. So these individual clones begin to diverge from each other and evolve independently. And as they do so, the cancer ends up having a unique natural history in each patient. So in this view of the world, genes aren't driving cancer. They're shaped by selection pressure. There's no driver mutation because they're all being shaped by selection pressure. So it may sound like a subtle difference. It may sound like semantics, but it's really not, because when you change the worldview from the oncogenic hypothesis or the oncogenic paradigm of cancer to the chromosomal instability or evolutionary paradigm of cancer, you get something very different. So the driving force of carcinogenesis in the oncogenic paradigm is the oncogene, which everyone who's done high school biology knows that the oncogene is the accelerator and the tumor suppressor are the brakes. Tumor suppressors are the brakes. And so what happens is a cell that has oncogenes expressed has its accelerator stuck. and if the tumor suppressors are mutated in that cell as well, the brakes are cut and the tumor just keeps progressing. This is how we've learned about it. Now, in the evolutionary paradigm of carcinogenesis, it's quite different. The initiating event is the initiation of chromosomal instability, which is analogous to releasing the handbrakes in the car on a hill. Chromosomal instability specifically means the gain or loss of chromosomes from one division to the next. This happens in cells that are pretty much every single tumor cell, and pretty much every single cell in which this happens is a tumor cell. So it's both a necessary and sufficient condition for carcinogenesis. Once that process gets rolling, because of unrepaired DNA double-strand breaks, often followed by checkpoint defects, often followed by chromosome misaggregation, that chromosomal instability process is self-sustaining. And so the car just keeps rolling down the hill. So that's the difference between the two paradigms. Now, that sounds kind of a little bit, again, it sounds subtle, but it's actually very, very significant, because in the chromosomal instability paradigm, what it would take to demonstrate that an agent is carcinogenic, any agent, is not whether or not it has an oncogene. For example, sunlight doesn't have an oncogene. And in the oncogenic paradigm, the argument is made that the radiation from the sunlight is mutating specific genes, oncogenes, and those mutant oncogenes drive cancer. In the chromosomal instability paradigm, the argument is that sunlight does something quite different. It generates chromosomal, UV light generates unrepaired DNA double-strand breaks. This is well known. And in cells with checkpoint defects, chromosome misaggregation eventually leads to chromosomal instability, which is the continuous process of gain and loss of chromosomes, and that ongoing somatic Darwinian evolution that's driven by this process of genetic diversity that's being created as a result of the chromosomal instability. So importantly, the evolutionary paradigm of cancer is hit and run. Once the insult is in place, the cancer will just keep going. The oncogenic paradigm is not hit and run. If you take away the oncogene, the cancer should go away. My own background is in cancer drug discovery and development. I've spent 20 years in that process. I was a discovery project lead for multiple programs that were in discovery and in development in big pharma. And I can tell you, I personally run programs where we knocked the oncogene out, we knocked down the functioning of the oncogene, and the cancers did just fine. Thank you very much. There's reams and reams of data that some of which hasn't even been published that shows this. And in fact, there's plenty of published literature that supports this as well. We had many manuscripts that never saw the light of day because the result was a non-result. We would knock the oncogene down and not see the effect we were looking to see. So where I'm going with that is that when you look at this from the chromosomal instability paradigm, all of this begins to make sense because you have the initiation of DNA double-strand breaks followed by chromosome misaggregation, which leads to an ongoing process of instability. Now let's talk about oncogenic viruses for a minute, shall we? So in oncogenic viruses, there's 7 viruses that are known or acknowledged by people to be cancer-causing or oncogenic. And these 7 viruses, all of them are, each and every one of these is something that causes a set of insults that are very predictable. And that set of insults is initiation of DNA double-strand breaks, DNA damage repair, in induction of DNA damage repair pathways, and then also disruption of DNA damage repair pathways. And then from there, we have chromosomal instability and microsatellite instability, which are 2 different things that happen. Chromosomal instability and micronucleus formation, which are 2 things that happen. And these two kinds of markers are typically associated with, as we discussed, the process of somatic Darwinian evolution within tumors. So every single one of these 7 viruses hits all 3 of these columns. We went through the evidence supporting that in a previous episode. And in addition, we'll have a number of further resources on this. We're planning to do some Substacks. We'll write this up with the references. There are also tweet threads on this if you're interested to learn more. But each one of the known oncogenic viruses causes carcinogenesis through a set of mechanisms that is very clearly defined in the sense of being associated with the downstream changes that lead to carcinogenesis. So let's move on to SARS-CoV-2. Now, SARS-CoV-2 infection leads to DNA double-strand breaks. People have looked at this in a range of different model systems. So there's multiple papers that show this. I'm going to show you one of them. We did this last time. I'm going to go over it real quick. What you're looking at here is a Western blot. So you're looking at the protein levels. Gamma H2X is a protein that binds to the end of DNA double-strand breaks. So the actin here is the loading control. It's just to make sure that everything is loaded the same way. And the gamma H2X levels, as you can see, in this particular case, are relatively low in the uninfected cells. Cells infected with SARS-CoV-2, the DNA double-strand break levels go up. On the right-hand side, you can see quantification of this. It's a pretty strong effect. So here's another paper that shows the same thing. This time they show it using microscopy. So what they've done over here is they've looked at this using, on the right-hand side, using protein biomarkers, which are basically gamma H2X, which we discussed before, as well as a counter stain for the presence of the nucleus. So this is immunohistochemistry. It's a process where you bind the antibodies onto the cells. So what you're looking at there is the nuclei of the cells in that sort of dark shade. And wherever you see these dots, that's where the gamma H2X is being bound. So you can see that in the controls, you don't see it, but in the treated arm, you see some pretty strong induction again of gamma H2X. Here's a third paper, again, looking at this with double-strand breaks. And you can see, again, they've quantified, in this case, gamma H2X. And they show across the board, an induction of gamma H2X. So this is a bit dependent on the one marker, gamma H2X. And it's kind of, we've looked at 3 papers so far. One showed it with microscopy, 2 showed it with microscopy, 1 showed it with Western blots. Here's a 4th paper where they've broadened this out a little bit. They are looking at gamma H2X again by Western blotting on the left. And then with immunofluorescence on the right, top right. In the bottom, they have the COMET assay, which is the gold standard for DNA double-strand breaks. So the COMET assay, basically what you do is you take these nuclei and you run them through an agarose gel. And so what happens is that the nuclei, if they have intact DNA, they just run just fine. And you can see that over here in the panel on the left. They migrate through the agarose under the influence of an electric current, which is called electrophoresis, many of you are familiar with. And you don't see anything leaking out of them. But if the nuclei contain broken DNA, then that'll leak out of them, which you see in the right. And these are called comet tails. You can quantify the comet tails and you can actually measure them. And that gives you a measure of how much DNA damage you're seeing. And it turns out, again, in the presence of SARS-CoV-2 infection, you get a strong induction of DNA damage in this case via the comet assay. So there are multiple papers on this. It's not very controversial at all. SARS-CoV-2 causes DNA double-strand breaks. So you could say, well, so what? Like DNA double-strand breaks? Well, it turns out that pretty much anything that's carcinogenic or nearly every single carcinogen causes DNA double-strand breaks. And the vast majority of DNA double-strand break agents are carcinogens. So that right there is bad news. It's not a one-for-one. There are things that make this one change and aren't carcinogenic, but your odds of it being carcinogen just went up quite a bit. So is this an artifact? Like, what are we seeing? So multiple papers have studied this in depth, and they've gone far enough to show that it's actually a mechanistic thing. And there are a couple of different ways in which this is happening. There's a protein called CHEK1 that gets depleted, which is a DNA checkpoint protein. You have nucleotide shortages, DNTP shortages, which are being caused by the replication of the virus. Because the virus is sucking up the nucleotides for its replication that the cell actually needs. And what this does, and we'll talk about it in more detail soon, is that it creates replication forks hanging around. So cellular DNA doesn't get replicated in a timely manner. DNA replication stalls for the cell. And when that happens, the odds of a double-strand break go up. So not only that, but as you can see, there are changes being made to the DNA damage signaling as well, such as the loss of CHEK1, which is the DNA damage signaling checkpoint protein. So here's another paper that talks about this for SARS-CoV, the closest analog of SARS-CoV-2. Apparently, this is a family thing. SARS-CoV as well causes replication fork stalling. And then those stalled and collapsed replication forks for SARS-CoV-1, not SARS-CoV-2, lead to DNA double-strand breaks. So it's all very plausible. And we're seeing robust DNA damage across the board with SARS-CoV-2, most likely by this mechanism. So at the same time, SARS-CoV-2 also causes micronucleus formation. As we discussed, when you have DNA double-strand breaks followed by defective checkpoint arrest and continued division through the cell cycle, broken fragments of DNA, and we talked about this in the previous episode, broken fragments of DNA cause chromosome missegregation typically, and that chromosome missegregation can lead to micronuclei forming. Those micronuclei are usually small chunks of DNA that are hanging out with their own nuclear membrane. So they're kind of like, as the name implies, micronuclei hanging off the main nucleus in a cell. A eukaryotic cell should have only one nucleus, not one big nucleus and a little nucleus like this, right? So those micronuclei are markers of chromosome missegregation. It turns out that the micronuclei are both diagnostic for and causal in chromosomal instability. So once you have micronuclei, that process of chromosomal instability is locked in because it becomes difficult for chromosomes to segregate, cells to segregate the chromosomes properly in the presence of micronuclei. So here, people were looking at buccal mucosa cells of COVID-19 patients. So they were swabbing the cheeks, and they found micronuclei in there. Here's another paper where they were looking at a different set in the tongue. So these are two different papers looking at micronuclei. And again, you see robust induction with strongly significant p-values of these kinds of micronuclei being formed in the mouths of people with SARS-CoV-2 infections. Here's a third paper that's looking at this time in syncytia, which are formed by, so SARS-CoV-2, one of the many nice things it does to us is it causes the cells in our body to fuse, it causes syncytia. And in those syncytia, often you'll see micronuclei formation, which is what's seen over here. This was done looking just at the spike protein. So syncytia induced by SARS-CoV-2 infection have an elevated level of micronuclei present in them. Now, when micronuclei are formed, as we talked about in the last episode, an internal signaling pathway kicks in. So when you get micronuclei, you have DNA double-strand breaks. That kicks in the DNA damage response, but there's an entirely separate pathway called the cGAS sting response, which essentially is part of the immunological response of the cell that causes micronucleated cells to be swept up and cleaned up. So that kind of garbage collection of cells with micronuclei happens not in a cell-autonomous way, but through the immune system. And so cGAS sting signaling essentially promotes downstream immunological clearance from the innate immune system. Now, SARS-CoV-2 micronucleus formation triggers that cGAS sting and DNA damage response. This is something that SARS-CoV-2 has in common with other viruses as well. In the last episode, we talked about how other viruses also do this. But as you may imagine, SARS-CoV-2 also disrupts DNA damage signaling. So this is a paper that we looked at previously. In this paper, we discussed a few slides back, we saw that SARS-CoV-2 was inducing DNA double-strand breaks. The authors in that paper went a little bit further and they looked at various different components of the cellular DNA damage signaling checkpoints, such as the enzyme DNA PK, ATM, and ATR. These are all components of DNA damage signaling checkpoints. There's some subtle nuances between what exactly they're doing in the DNA damage checkpoints, which I won't get into today. But the point is that these are proteins that are required for downstream DNA damage responses of various different types. And what we can see over here is that in the presence of the virus, the activation of these checkpoints is altered. So there's altered DDR activation that's happening as a result of SARS-CoV-2 infection of cells. Again, if you already induce DNA double-strand breaks, if the virus induces DNA double-strand breaks, having the virus then go ahead and disrupt the cell's own response to that is, you can imagine, not good news. There's a whole machinery inside these SARS-CoV-2 viruses that is aimed at disrupting surveillance of micronucleated cells. The viral proteins downregulate the DNA damage response, they suppress apoptosis, and as I'll show you, they also interfere with cGAS sting signaling. So here are a bunch of references. And again, these references will be spelled out in further in the work that we will be sharing with you on Substats and in reviews. Some of these have also been fleshed out in the tweet threads. But so what we're looking at here is we're looking at the downstream machinery that exists in SARS-CoV-2 cells to facilitate the escape of cells that facilitate SARS-CoV-2 virus, to facilitate the escape of cells that have micronuclei or DNA damage. So all of the machinery that's involved in the cellular machinery that's involved in the surveillance of cells like this is actively being suppressed. So viral NSP15 protein binds to cellular PRB and targets it for degradation. P53 is downregulated as well. Now, people who talk about SARS-CoV-2 from the oncogenic perspective will reference these exact papers. And that's just a different paradigm of thinking about it. As I discussed, we prefer to discuss it using the chromosomal instability paradigm. So in the chromosomal instability paradigm, what's happening is SARS-CoV-2 is disrupting the management of the DNA damage response at the cellular level. Very often, the DNA damage response will trigger apoptosis. SARS-CoV-2 interferes with that apoptosis. So it goes after that. It turns out, to no one's great surprise, if you have SARS-CoV-2 infected cells and they contain DNA damage, those DNA damaged cells continue to proliferate. So what you can see over here is the N protein, when it's overexpressed, causes reintroduction of cells into the cell cycle. So once you have DNA damage, you've got micronuclear formation, those cells continue to cycle. It doesn't take much thinking to realize that this is probably pretty damn advantageous for the virus, because if these are cells that are factories that are producing SARS-CoV-2, then it's not in the virus's interest, or to put it differently, the virus can get a fitness advantage from making sure that those cells proliferate. But in doing so, it's not doing the host any favors. As I mentioned, multiple SARS-CoV-2, not one, but multiple proteins interfere with the cGAS sting signaling pathway. So once you have micronuclei formed, there's multiple proteins within the virus. There's a whole panoply of machinery that's being deployed to make sure that those cells continue to cycle and that they evade elimination by the immune system through the cGAS sting pathway. So all of this sounds very diabolical at one level, but hey, looking at it from a different perspective, the good news is it's doing all the stuff that other textbook viruses do, right? Textbook carcinogenic viruses, that is. So SARS-CoV-2 generates the same cellular insults as viruses that are recognized to be carcinogenic. It also, by the way, for good measure, infects T cells and kills them. Here's a paper from several years ago that showed that T cells were being infected directly by SARS-CoV-2. I'm going to go through this a little bit quicker because this is a level of detail that is sort of stacks on top of what we've seen. We've already seen that SARS-CoV-2 is capable of producing these cells and is capable of allowing these cells to escape innate immune system elimination and DNA damage checkpoint elimination. Now, the third layer of defense, which is the cell's adaptive immune system, is also impacted by the virus. So that's three lines of defense that the virus is getting through after having produced these cells that are already chromosomally unstable. And actually, once it infects these T cells, it kills them. That's been shown in this paper. There's a number of papers of this nature that show this. It's been well demonstrated that SARS-CoV-2 suppresses T cell signaling as well. So everybody who says your T cells will save you with SARS-CoV-2 should also remember that they can't save you, they're dead. So here's a paper that shows that when you transfect with a homologue of a SARS-CoV-2 homologue of a peptide of NSP, of the peptide NSP7, you end up with T cell proliferation being downregulated. So NSP7 is involved. The inference is that NSP7 is involved in regulating T cell function during infections. It's well known that T cell populations are depleted in severe COVID. The Merck manual lists SARS-CoV-2 infections as a cause for lymphocytopenia. But people will often say, well, that only happens in severe cases. It turns out, as this paper shows and other papers have shown as well, that also happens in mild cases of COVID. The paper talks about severe COVID, but if you look at the mild, the examples where they're looking at mild COVID, which is the stuff in blue, you can see that often T-cell exhaustion is not as impacted, but other things are. For example, T-cell activation and cytotoxic T-cell numbers changing. So you're seeing a lot of the same changes in mild COVID as you see in severe COVID. As I mentioned, the Merck manual actually lists SARS-CoV-2 as a lymphocytopenic agent. If you look on the right, this is a different paper, right from 2020, March 2020, that shows in both non-severe and severe cases of COVID, you see lymphocyte numbers that are outside, particularly T cell numbers that are outside the normal range. So go figure. And this again, this is just one example. That I've listed multiple references here. Again, when this makes its way to publications for us, we'll be putting all those references in. But the point is, it's not difficult to find examples like this. And this is a key point, that SARS-CoV-2 also ablates the T cell population in many cases. Here's another paper that shows this with severe symptoms as well as mild symptoms. You see SARS-CoV-2 infections causing levels of, as you can see, this is the healthy control number, 2.65 plus or minus 69 for lymphocytes. And here's what you get with SARS-CoV-2 infections. So here's another example, DNA damage in lymphocytes with both mild and severe symptoms. There's also extensive innate immune system suppression. Separate from suppressing the C-gas sting signaling, for good measures, SARS-CoV-2 also screws with, as they say in French, your cytotoxic NK cells, natural killer cells, your dendritic cells as well, both of which are required for your innate immune system to perform surveillance. So there's just extensive perturbations in white blood cell populations and signaling with mild cases, with severe cases, as well as in the long COVID setting. In addition to that, SARS-CoV-2 also causes chronic inflammation. This has been very well documented. I'm not going to go into any detail because, it's easy enough in terms of a PubMed search to go find these papers, but it's not controversial that SARS-CoV-2 causes chronic inflammation. So when you look at it that way, there are actually many different mechanisms separate from the carcinogenesis that happens from that 3 or 4-step process that I described earlier, which is DNA double-strand breaks, suppression of the DNA damage response, induction of chromosomal missegregation, followed by chromosomal instability / micronucleus formation. That was cell autonomous. Now we're taking a step back and we're looking at it at the tissue level. And the point is, at the tissue level, there's all kinds of other things happening as well. So there are many plausible mechanisms for SARS-CoV-2 to promote cancer progression. This is taken from a review that makes this point. It's not my point. So that's great, not so good, right? But the point is that this could be theoretical, right? If it's just in, if it's just in 1 or 2 tissues and those tissues are not replicating, then we have nothing to worry about, right? Then this is just a theoretical thing. Unfortunately, the virus infects nearly every tissue. So SARS-CoV tropism is extremely broad. You can find SARS-CoV-2 everywhere. Whenever people look for it, they more or less find it in infections. And even when people have looked at mild infections, they find the virus in places they weren't expecting to find it at all. So the notion that it's just an upper respiratory infection, 2020 called and wants its definition of the virus back. In autopsies, patients who died of severe SARS-CoV-2, they find it everywhere. So this is in each column, you have a different patient. And these are autopsies where they went and did PCR to ask the question, where are we finding the virus? And what they were able to find was that the virus is present very broadly in different patients, not in all of the same tissues, but kind of in a stochastic random way scattered throughout the body for each patient. The bars indicate the presence of replicating virus. So they can look for replication intermediates, subgenomic RNA. And so wherever you see the bars, that's where you're not only seeing that level of viral RNA, but also it's replicating. And as you can see, replicating virus has been found in places like the optic nerve or in inside the brain, or in the small intestine, for example. Some of these are replicating tissues. Not only that, but SARS-CoV-2 infections directly lead to a wave of regenerative proliferation. So it's been shown that in human samples, in human clinical samples, when you have small intestinal infection with SARS-CoV-2, you get a mucosal infiltrate of activated CD8 T cells. Great. So, so what, right? But so first of all, you're seeing that if you're getting activated T cells in there, that means something's going on. So these guys went and looked at this in a little more detail. And what they found basically was that the number of Ki67 cells, or actively proliferating cells, went up. So what you can see over here in that teal color is proliferating cells. These finger-looking things are the villi in the intestine. And what you can see is in the control, there's a small number of proliferating cells. On the right-hand side, in the SARS-CoV-2 infected cells, first of all, the villi look really messed up, right? The structure of the villi has changed. You can see the structure with both the marker DAPI, which marks the nuclei, and the marker EpCAM, which is an actual marker for the epithelial marker showing you the villi. What you can see is over here, in contrast to this regular sort of finger-like structure in the control, patients with SARS-CoV-2 infections in the small intestine have villi that are damaged. And in those damaged villi, you can see with Ki67, you can see the teal color. Those nuclei show replicating cells. Ki67 is a marker for proliferating cells. So you can see a pronounced increase in the replicating cells. So these folks went ahead and they built a little, paper model of this little drawing of it. And what they're showing basically is that when the virus shows up and it starts killing off cells, there's a wave of regenerative growth from the intestines. So you could say, well, gee, that sounds great. That's all dandy, right? So all the damage is being repaired, nothing to worry about. Well, it turns out there are other pathogens that cause this sort of cycle of killing off cells with rapid regeneration. For example, Helicobacter pylori, which is carcinogenic in the stomach, does the exact same thing. It doesn't take much imagination to see why having a virus that's actively damaging DNA in the presence of a whole bunch of proliferating cells, which, by the way, the virus is inducing, is not going to end well in many cases. So that combination of sort of both being present in proliferative tissue, driving proliferation, and then also causing DNA double-strand breaks in those proliferating cells and disrupting downstream signaling. If that's what's happening in the crypts, and we don't know that for a fact right now, but you can put two and two together and say, if that's what's happening, it's really bad news. We know the individual pieces are true. We know that SARS-CoV-2 causes DNA double-strand breaks. We know that SARS-CoV-2 disrupts all manner of downstream signaling to arrest it. We know that SARS-CoV-2 infects and kills cells in the villi. And we know that as a result of that, there's a wave of proliferation that follows. Missing paper is the one that shows that all of those things happen at the same time. But if all of those four things happen at the same time, you have a smoking gun for carcinogenesis. So if I was a betting man, I would make a bet that you're probably seeing this in the crypts and in the villi of people with SARS-CoV-2 infections when the infection goes to the intestine. You're probably seeing this with people with long COVID who have intestinal involvement. So the pieces exist. Someone has to go and literally, put them together in a paper. But it's a plausible hypothesis, and it's one it's difficult to just dismiss it out of hand because all the pieces exist and other pathogens that have those pieces are in fact carcinogenic. So let's zoom out a little bit and let's talk about the checklist of our oncogenicity. We talked about this the previous time with the other 7 viruses that are known to be carcinogenic. The first question is, does the virus cause persistent infections? We saw that all of those 7 viruses that we discussed last time do in fact cause persistent infections in dividing tissues. Yes, they cause infections in dividing tissues. And then we showed that cells infected with those viruses experience DNA damage that doesn't resolve and micronucleus formation. For each of those 7 viruses, we showed that. We also showed that there's, to varying extents for those viruses, all of these suppression of all of these different downstream pathways, lines of defense, both at the cellular level, such as DNA damage response and induction of apoptosis, and then at the cell independent level, which is the suppression of cGAS signaling, and finally the suppression of the immune system generally. We've seen that with all of those other viruses. We've also seen with some of the other viruses, and we've also seen for some of the other viruses, but not all of them, chronic inflammation. The important point is that the first three larger bullet points you see in all seven of those other viruses. And then in some of the others, you see this fourth bullet point. So how does SARS-CoV-2 do? Just to sum up what we saw today, it cleans up. It really cleans up. So I would be surprised if it was not carcinogenic, based on this. Again, it's not been proven directly, but all of the pieces exist. And from a mechanistic standpoint, if you're using deductive logic, you would focus your attention on ruling in or ruling out this hypothesis on the basis of deductive logic. As I said, for example, in the crypts, all the elements are present. We should be able to demonstrate that there is direct induction of micronuclei in those crypt cells. If we see micronuclei in those crypt cells, somebody goes out there and does that paper in clinical samples, I think we have definitely something that's a long part of the way towards being a smoking gun. So that wraps it up for today. Again, we put forward a plausible hypothesis that deserves consideration that SARS-CoV-2 is actually carcinogenic on the basis of the fact that it makes a bunch of changes to cells during infections that are completely consistent with what 7 other viruses do that are known to be carcinogenic. And just by the way, I can't think of a single virus, and I invite people to share with me examples of viruses that do all of these things and are not carcinogenic, or even pathogens that do all of these things that are not carcinogenic. So in the context of the general mechanism by which carcinogenesis happens under the chromosomal instability paradigm of cancer, this is what you would expect to see. You see it with the 7 viruses that are known to be carcinogenic, and you see it with SARS-CoV-2. So as they say, you do the math. I think the odds are stacked in favor of SARS-CoV-2 being carcinogenic. Now, that being said, it's not definitively proven, right? And as I said, one way to definitively prove it is to prove it with deductive logic, to go in there and show that these kinds of changes are happening in tissues that are proliferating. That proof is pending. There's other things you can do with deductive logic to shore up this case. But you could also ask the question, well, why don't we just wait to see what happens with the data and see what happens with rising cases in cancer and whatnot, right? A counter argument often made is, well, it's not like it's not like we have large numbers of rising cases of cancer. So what's going on there? In the next episode, we'll talk about epistemologically, how easy is it to prove that a virus is carcinogenic based on historical standards? And what are the costs? What's the cost benefit of doing that? But that's it for today. And we can pause here. And I'll take questions from the extensive audience.
From the audience, yeah. I think it's a very, very interesting and I want to say alarming, but I think hopefully motivating narrative that makes a lot of sense. Now, there have been a couple of papers suggesting or playing with the idea of SARS-CoV-2 being oncogenic I think in the traditional sense of oncogenic viruses. What's your take on some of those discussions as they occur in the sort of old paradigm?
So, I mean, in the old paradigm, the way in which it's framed is basically in terms of oncogene. So pretty much every oncogenic virus except for HCV has an oncogene in it. HCV doesn't. It causes chronic inflammation, but aside from that, every other one has an oncogene in it.
And so when they talk about SARS-CoV-2 in that context, are they asking whether any of the genes contained within the genome of SARS-CoV-2 could be an oncogene, or so that's
and there are not. There are not. So everybody who says, well, it's not oncogenic, they're like, there's no oncogene. Sure, it's not oncogenic in that way, if you use that definition. But if you talk about carcinogenic, it causes DNA double-strand breaks, causes chromosomal instability. It suppresses pretty much every single downstream mechanism that would prevent cancer from forming as a result. So what's your money on is how I look at it.
It's interesting. I think there's going to end up being some need to have people grasp what is going on in terms of benefit to the virus. I think it's not the case necessarily that causing cancer per se is, advantageous from the standpoint of the virus. I think what you have in the scenario you've laid out tonight is it's essentially using up the resources in the cell to make copies, which is indirectly causing the replication forks to collapse and giving you the double strand breaks, which initiates the process. And then it makes sense for it to suppress host
Anything
control
downstream.
mechanisms,
Yeah.
right? So it's not necessarily trying to, it's not necessarily causing cancer per se. It's what it's doing for its own benefit is indirectly causing the double strand breaks, and then it does want to suppress, to use sloppy, language, but it is to its benefit for those responses to be suppressed that would otherwise terminate the cell and then obviously kill the.
Yeah. I would put maybe a little bit differently. It is very
Yeah,
much
go ahead.
under this hypothesis, it's very much causing cancer, but it's not doing so because it quote unquote wants to, if we were anthropomorphizing it.
Yeah,
It's doing so because it wants to replicate as much as possible.
exactly.
So from an anthropomorphic perspective, which is deprecated in the evolutionary world,
Yeah.
as we both know.
But it's easy
But
to
if we were
put it into accurate
anthropomorphizing
language.
it, yeah, then we would say it's not causing cancer because it wants to. It wants to replicate as much as possible, and cancer is a byproduct. To put it differently, in a cleaner evolutionary language, it's not gaining any fitness advantage from causing cancer, but neither is it being disadvantaged at all.
Yeah.
And it does gain a fitness advantage from suppressing the downstream mechanisms that would make those cells that already have carcinogenic changes in them from being cleaned up or being arrested.
And that's somewhat where I wanted to go with it, in the sense that, some of the thinking about viruses will attenuate or will become less virulent because they need to, not do damage to the host lest they use up all their hosts. That itself is problematic from an evolutionary theory standpoint. But these kinds of changes that take a while for the negative impacts to be seen in the hosts are not, there's essentially no selection
Not covered.
against
Yeah.
them.
It's
So
not covered.
all
Not
of
covered
this
by
can be
your
all
God
of
or
this can
mine.
happen,
Yeah.
right? And it makes sense from the standpoint of sort of the immediate benefit to the virus, with essentially no long-term cost to the virus. It's already moved on
Right.
to new hosts. So
Right.
we need to be very clear, I think, not you and I, but when we're thinking about, negative impacts or virulence, there's this prolonged effect that isn't even part of the sort of standard incorrect, but standard thinking on attenuating viruses. This kind of thing can absolutely happen without real detriment to the virus and be accentuated.
So the virus is under no contractual obligation to attenuate.
No.
Not only that, but one cannot even conceive of a mechanism by which a virus would attenuate for something like carcinogenicity, to your point. There's not even a plausible, like even if I was hand waving, no matter, how many beers I drank or weed how many joints I smoked, I couldn't even come up with something that would be remotely plausible.
Yeah.
So yeah, this is sort of where you really have to invoke, if you're invoking magical thinking or you're invoking, a divine intervention, you really have to stretch it to say that this is not, if it's a problem, you really have to stretch it to say, oh, it's a problem now, but it may not be a problem in the future. No. The problem will only get bigger in the future.
Yeah, and I think that sort of came up, I think, last time, which is there are plausible scenarios in which this gets worse over time. So, for example,
Yes, and we'll discuss that. Yeah.
Increased persistence or tropism to those tissues in particular that are dividing rapidly.
So, and in fact, it operates at two levels. One is, if this is happening, it almost for sure isn't visible right now, because it takes decades for cancer to form. And by the time it's visible, a lot of that damage will already have been locked in. As a second point, what you just said, which is during those 10 or 20 years that it takes for such an impact to be recognized as being, oh, it's a problem, the virus may have moved a good distance evolutionarily and maybe actually much better at doing this than it is today. So in all respects, the problem can only get bigger from where it is now, if it exists. And so this is exactly one of the situations where wait and see is just about the worst thing you can do.
Yeah, and one of the scenarios that we've been playing around with that I'm increasingly interested in is a shift in life history strategy, so to speak, of a sort of rapid replication and high acute virulence, which we saw early in the pandemic, to potentially more persistence, movement to other tissues that on the short term and using the metrics that we continue to use, which is deaths and hospitalizations and ICU and so, usage and so on, would look like an improvement, but really it's taking up residence in places like the gut or kidneys or anything else that maybe
Yeah.
this could become worse over time. And there are scenarios in which that would make perfect sense from the virus's standpoint.
Right. I mean, a lot of this, in order to buy into a lot of that framework, it requires sort of a commitment to the theory of divine intervention, because there's nothing in the history books, and there's nothing in the biology literature that says that that's how it has to go.
That it would
It
attenuate.
may end up there. Yeah, it may end up there. But it's exactly like saying, hey, I walked down, the street in the Garden District in New Orleans at 11 o'clock at night, bombed out of my mind, and nothing happened to me. And therefore, I'm going to keep doing that because I've proven that it's safe. If you do something that's dangerous and disaster doesn't follow immediately, it doesn't follow that disaster cannot strike. And so the whole notion that repeated waves of the virus will lead to better outcomes because the last couple of years have, quote unquote, gone better, which itself is debatable. That's a very bold extrapolation.
Yeah, I think it's very much an unevolutionary view, which is that it's heading in a particular direction, and that once there, it necessarily stays there. There won't be further change. And I think it's just
Right.
totally.
Yeah.
I mean, the more you have
Yeah.
to dig into the variation and so on.
I think in my own personal life, the way I look at it is, I don't need to see the paper that says we have proven beyond a doubt that SARS-CoV-2 is carcinogenic. I have a pretty damn good clue that it's carcinogenic. I got COVID last month or 2 months ago, and we did everything we could to keep it out of our guts. Right? You don't want it to take up shop in your gut, and there are ways to do that. You and I have talked about that separately on our Substack blogs and so on. But you really don't want this thing taking up residence in your gut. In general, you want to get COVID as few times as possible. And in general, if you get it, you want to get it out of your system as quickly as possible. If you think it's a carcinogen, you will react to it in that way. Just in the same way as when you're out in the sunlight, you rub sunscreen on yourself. It's kind of the same thing. You use prophylactic measures to limit your exposure. It's not complicated. But if you don't think it's a carcinogen and you think it's just a cold, you're going to have a very different set of experiences with the virus today, tomorrow, 1 year from now, 5 years from now. And that cumulative sort of flirting with exposure with this virus is going to lead you to the same outcome that that the Australians and the South Africans experienced in the 80s, when all of a sudden they realized, tanning out in Bondi Beach or whatever is not a good idea because Australia had this sky high rate of melanomas, right? So it's one of those things where if you don't think that it's a problem now, you may find out later that you were wrong. And so if you assume it's a problem, then you take that sort of analogous to put on your just wear your damn sunscreen approach. You're going to be in a better place 5-10 years from now than if you're not. And if it turns out that everything was fine, great. Then you wasted a bunch of analogously you wasted a bunch of sunscreen.
I think that's another really important point, which is it's not going out into the sun once, right? It's a cumulative risk thing, and so repeated infection. You also,
I mean, to that point exactly. Like, do I think that SARS-CoV-2 will immediately strike you down with cancer, even if it is a carcinogen? No, carcinogens don't work that way.
Yeah,
they really don't work that way. You can smoke as many cigarettes as you want. You're not suddenly going to get a tumor bulging out the side of your neck, right? But there's a big difference between the cancer risk of someone who smokes 3 packs a day and chain smokes, and a big difference between that and someone who smokes a cigarette a day or smokes a cigarette every other month. I'm not saying that there's any safe level of cigarette exposure. There's not. But obviously the two sets of lifestyles lead to 2 very different outcomes in the long run.
I think you raised another important point about it getting into the gut. in one of the more recent waves, people were pointing out, okay, there's a lot in the wastewater, a lot being detected in wastewater, but not so much, in terms of hospitalizations, severe respiratory uh, outcomes, and as though
Yeah,
that's a good thing, right? That's
people were excited about it. Yeah,
they were excited about it, meaning that see how mild it is. There's lots of virus, but not a lot of disease. I guess I looked at it and said, yeah, we don't really want a lot of it moving to the gut. Like that's
yeah,
along the
it's
trajectory
not.
we're talking about, right? That
Yeah, I think in general, it's just incredibly reckless um to push this narrative that somehow, deaths and hospitalizations are the only metric we should be concerned about. With the rapidly evolving virus, infecting people, once a year, twice a year, once every other year. Uh, there should be management that is aimed at potential downside risks. In general, in our lives, we always think about what can go wrong, not what can go right in terms of risk management. So I think the whole approach is just incredibly reckless. But I think you've highlighted another another way in which it's reckless.
but one of the things, like, I mean, this is the last question I'll have on this one is about carcinogenesis that's caused by viruses, and I guess there's two ways to kind of look at it. Like, how are there a lot of cancers that are likely to actually be the result of a viral infection that we don't currently recognize as having a viral origin? And the flip side being, are there a lot of viruses that have the potential to do this? I'm just thinking about the mechanism that was outlined for
Yeah,
SARS-2. It's not incorporated into the genome. It doesn't contain an oncogene, but the
yeah,
depletion of the nucleotides, right? That could
yeah.
be true of many
So
viruses, right?
I'm not a virologist and I'm not a clinical oncologist. What I have done for like a very large chunk of my career is I've worked on drug discovery and development for cancer. And what I've seen, just bluntly put, I'll be blunt about this,
Mhm.
is the entire community was thinking about the problem wrong 2 decades ago.
Mhm.
It was just very pervasive. The oncogene hypothesis. It was like a sickness, right? You had AACR after AACR, American Association of Cancer Research, where people would present paper after paper after paper on biochemistry that was not reproducible, that was not reproducing, creating these narratives of these signaling pathways that were being switched off and switched on, and framing what was actually happening at an evolutionary level as biochemical adaptation.
Mhm.
And they were force fitting this deterministic view onto a stochastic system. So the entire field was basically wasting its time. If you go back to the papers that have been published in cancer from maybe the 70s or 80s up to, I would even say, 2005, 2010, there's just a huge chunk of that work that isn't going to lead anywhere. Over time, the field has kind of recovered and has sort of rethought it. But it was a paradigm shift. And I think that paradigm shift is visible in the cancer field today, although you still see papers presented talking about oncogene addiction. If you look at a graph of the number of papers that talked about oncogene addiction over time, it kind of went up really fast. Hockey stick stayed there for a long time and then sort of slowly started declining. And as I say, a paradigm doesn't change until the people who were trained in that paradigm retire. It
Mhm.
takes a long time. Where I'm going with that is that I think something similar will have to happen in viral oncology, because viral oncology will have to go back and say, gee, were we thinking about this process wrong? Were we thinking about viral oncogenesis wrong? If we now wear the chromosomal instability hat, by the way, the people who are working on cancer outside of viral oncology are now doing, if we now wear that hat, what does it do? And I suspect, long-winded answer to your short question, I suspect that we'll find a lot more viruses are capable of causing cancer. But I personally, like, I don't want to wade into that. I'm
Mhm.
invested right now in one virus. There's probably more
Fair
than
enough.
this.
Fair enough.
We have, and I think just to make this point, I think there is no worse virus to be carcinogenic right now than this one, because this is an ongoing public health crisis and everybody is getting it, once a year, once every other year, twice a year. There's not another virus I can think of that's happening as often.
I would almost argue there's never been, I mean,
There's never been,
with
probably.
this many infections, this many hosts, global travel, this rate of evolution, it's pretty
Right.
unprecedented in that regard.
So even if it was a weak carcinogen,
Yeah.
we are taking the wrong approach to it.
Yeah.
So in a certain sense, like, um, the precautionary principle says if you don't know what the downstream risk of something is, you measure your risk and you control your risk and you control your exposure. We're doing the exact opposite with this thing. So we're kind of like we're all like those radium girls in those watch factories, just licking our paintbrushes right now. And I think, it's been proven over and over again that human beings can be um, pushed into situations where they took on risks that they weren't expecting, like the Dutch drinking out of their lead cups in the 17th century, or the girls working in those factories, licking their paintbrushes and then coating radium onto the watches. People
Mhm.
can often get exposed to stuff that they didn't know was harmful, and that's in modern times. You know, theoretically, like they tell you these stories, and then they say, "Well, we're so much better now,"
Hmm.
right? Well, are
Yeah,
we?
but not really. Well, on that note, and I think just a reminder as to the follow-up of this episode, we'll be looking at what it would sort of involve to actually show that this is really is a carcinogenic virus. And I think just to kind of preface that uh, with that's not something we want to wait to find out. I think is kind of a key message even from tonight. So, but it's going to be difficult to do. I think as it is for many things, and has been historically. So, you know, do we learn from those prior examples or not? I think is another important question. Seems like so far not so much. But in any case, very interesting stuff. And we'll see everyone at the next episode.
Thanks.